revolutionary tool decodes genome folding, unveiling DNA's 3D blueprint. Newswise, byline, Laura Red Eagle. Newswise. In a groundbreaking discovery, researchers at the Wisconsin Institute for Discovery have cracked the code on a mind-boggling packaging challenge. A sophisticated computational tool unveiled in a recent study published in Genome Research /genome.cshLP.org/content/early/19/GR2799930-124 abstract allows scientists to unravel the mysteries of how genomes compact themselves within the minuscule space of a cell nucleus, how they reconfigure during various biological processes, and how these transformations impact gene expression and disease susceptibility. As cells evolve and differentiate, their genomes undergo massive rearrangements, occurring at different developmental milestones, disease stages, and other temporal junctures. Gene expression requires precise orchestration, as alterations in the three-dimensional structure of the genome have been tied to alterations in gene activity that underpin diseases such as cancer and genetic disorders. Fascinated by this enigma, Sushmita Roy and Da-In Lee, graduates of the University of Wisconsin-Madison, devised a computational technique known as tree-guided integrated factorization, TGIF. This method employs a machine learning strategy called matrix factorization to construct a mathematical representation of DNA folding. Roy and Lee envision TGIF as a means to comprehensively explore how DNA modifications across various dimensions affect traits ranging from hair color regulation to genetic disorders. One of the cardinal questions in mammalian genomics pertains to the intricate packaging of DNA within the nucleus, ensuring that pertinent segments are accessible for cellular reading while others are tucked away, contingent on context, explains Sushmita Roy, professor of biostatistics and medical informatics and WID faculty member. Historically, non-coding DNA was deemed insignificant due to its lack of protein coding capacity, which is essential for tasks like tissue construction, infection defense, and chemical reaction regulation. Nevertheless, researchers are discovering that non-coding DNA performs vital roles, such as directing or modulating gene activity and protein synthesis. What remains elusive is the exact correspondence between DNA segments and gene control. Investigating nuclear DNA packaging could hold the key to unraveling gene regulation in both healthy and diseased cellular functions, posits Roy. Past methodologies scrutinize these alterations across dual time points or conditions. The Roy Lab sought to devise a tool capable of monitoring changes over multiple time intervals and accommodating intricate architectures. Upon validating the analytical framework, they observed a robust correlation between substantial and minor fluctuations in 3D genome architecture and variations in gene expression, particularly in genes engaged in time-specific functions. They also explored the demarcations between densely interacting genomic regions and discovered that stable boundaries were frequently linked to genetic variations associated with disease. This constitutes a pivotal stride toward elucidating the genotype-to-phenotype relationship, a cornerstone in understanding organismal functionality under diverse environmental circumstances, asserts Roy. For instance, we identified that preserved boundaries were connected to single nucleotide polymorphisms implicated in cardiovascular disease. Equipped with this novel instrument, the WID team is furnishing a potent addition to the gene regulation puzzle, one that could aid scientists in not only deciphering how the genome operates, but also how its configuration influences our very being. Laura Red Eagle.